Hi everyone, today we are going to discuss about the another important topic in human reproductive system, that is about the oogenesis. Like a spermatogenesis. What is the oogenesis? The process of formation of matured female gonads developed in ovaries is called oogenesis. The next this is about the definition of oogenesis. Now we are discussing about the ovulation. The difference of oogenesis and ovulation. Ovulation means the breakdown of Graafian follicle to release ova is called ovulation. Ovulation means after the matured Graafian follicle, after the formation of the matured Graafian follicle. The next, the first releasing of ova. The first released ova is called as menarche during the period of the 13 to 19 years. So means adolescent age. To stop the releasing of ova, is called menopause. In the oogenesis process, there is a 3 steps is there. The first one is multiplication phase. Second one is growth phase. Next, third one is maturation phase. The 3 stages. The first we are going to discussing about the multiplication phase. The multiplication phase is started during the fetal development of the females. In the 3 stages, the first stage is here the multiplication stage. The second one is the growth phase. This is about the growth phase. The third one is the maturation phase. The first we are discussing about the multiplication phase. See here in the multiplication phase, we said that this is started in the fetal development. It is started in the fetal development. See here in the fetal development, in the fetal development, the number of the cells are there in the fetal development. Number of the cells are there. In this number of the cells, what happened? The certain cells means in certain cells become a larger. Why in the certain cells to become a larger? Because it is involving in the mitosis solution. These certain cells involve in the mitosis solution, then it become a larger one. This larger one is called as the oogonium. This oogonium. This oogonium is called the mother of the ooted or germ cell of the ooted. It is the 2n nature. The diploid condition. In this the oogoniums, what happened that this is developed into a, these oogoniums to form the one operator that the egg tube flagger. It is developed into a oogonium developed into a egg tube flagger. to develop into an egg tube flagger. This egg tube flagger, from the egg tube flagger, then it is formed into a egg nest. It is formed into a egg nest. In this egg nest, nothing but a mass of the ovas, a mass of the egg is called egg nest. In this egg nest, only one to develop, one is involving in the mitosis cell division, from which in the egg nest. One is certain cells, again egg tube flagger, egg nest. In this one egg, one oogonium to involve in the mitosis, then form a primary oocyte. But remaining, which is the ovas, is their egg nest, there is a to lose the to involve the mitosis. They are unable to involve the mitosis. Only involve the mitosis, then to develop it a primary oocyte. This primary oocyte also, it is how many? The 2n nature is diploid in nature. Nothing but the 46 chromosomes is there. 46 chromosomes is there. This primary oocyte to involve the meiosis 1. To involve the what? The meiosis 1. After involving the meiosis 1, then to form into a two unequal cells. Two unequal cells. Which is consisting the high amount of the cytoplasm, that is called secondary oocyte. That is called what? Secondary oocyte. Which is the low amount of the cytoplasm, which is called, that is called the first polar body or the polar body. From the oogonium to primary oocyte is called multiplication phase. Then now the primary oocyte to formation of the secondary oocyte, it's called the growth phase. Then now here the unequal cells are formed. One is a high amount of the cytoplasm is there, another scanty of the cytoplasm, that is called polar body. High amount of the rich amount of cytoplasm, that is about what? Secondary oocyte. Here the secondary oocyte is a end, because here the meiosis, so the chromosome become a half, means here the 23 chromosomes are formed there. This about the what? The growth phase. This secondary oocyte again to involving in the along with the polar body to involve in the meiosis 2. This meiosis 2, after the completion of meiosis 2, so what it is arrested in which phase? Metaphase 2. It is arrested in it is involved in the meiosis 2, but arrested in which phase? Metaphase 2. It is arrested in which phase? The metaphase 2. When it is complete the cell division, when it is mating with the sperm, it is completed the metaphase to that. So then after that, what happened? The secondary oocyte to involve the meiosis 2, then form the one ooted and unequal division again, and one polar body. Here the first polar body is there to involve the same as usual like secondary oocyte, then form the what? Two polar bodies. Two polar bodies. So here the entrance examination question, that one oogonium to produce how many ootids? Only one ootid. One oogonium, only one ootid. One spermatogonia, how many sperms? Or spermatozoa? 4. But in spermatogenesis, there is no polar body. Here the formation of the polar body. One oogonium, 3 polar bodies are formed. 3 polar bodies are formed. What is the work of the polar body? The polar body is the help to the high amount of the development of the cytoplasm in ootid. Cytoplasm in ootid. The ootid also the 23 chromosomes is there. But the ootid, which type of the oocyte is there? That is secondary oocyte is there. Which oocyte? The ootid is nothing but is a secondary oocyte. Secondary oocyte. This about that. Now in entrance examination, now they are asking what are the phases: multiplication, growth, and maturation. What are the development of the ootid? How the formation? The first is the one oogonium. First is the oogonium. Oogonium is a singular. Oogonia is a plural. Oogonium. Oogonium to primary oocyte. The primary oocyte to secondary oocyte, secondary oocyte to ootid. This is the formation, the way. Then next in that again oogonium is a 2n nature. The primary oocyte is a 2n nature. The secondary oocyte is a n nature. The ooted also N. The ooted is nothing but a secondary oocyte. Secondary oocyte. The next, how many polar bodies? The 3 polar bodies is formed. That where it is arrested, meiosis 2, metaphase 2 is arrested. This is about the spermatoogenesis. Now we are going to discuss what is the significance of the oogenesis. We are going to discuss significance of the oogenesis. We discussed already that one oogonium to produce the how many ootids? The one ootid or one ovum. Again, how many polar bonds? The 3 polar bodies is there. Already we said the polar body to provide the sufficient cytoplasm to the ootid or the ovum data. So what is the second one? During the first crossing, now over the variations are meaning there is a due to the first meiosis one, there is variations, the differences will be formed that in the child. The next it is the secondary oocyte of human, that is the A-lecital. A-lecital nothing but here the absent of yolk shock. Due to the absent of the yolk shock, it is called A-lecital. A-lecital. This is about the significance of the oogenesis. See here this is the just diagram of the secondary oocyte. See here the diagram of the secondary oocyte. It is a spherical shape or oval shape is there in the spherical shape that. See here in the secondary oocyte is covered by the membrane, which is covered by the plasma membrane. It is covered by the membrane that is about the plasma, the membrane. See here that plasma membrane, after that, which is the liquid position. What is the cytoplasm of the ova is called ooplasm. Ova is called ooplasm. In the ooplasm, number of the ovas is present. Number of ovas is present. Here in the ooplasm, applied nature, the nucleus is present. Applied nature of the nucleus is present that. See here, the ones this is the diagram. If you are observe that it is covered by the which plasma membrane. That membrane we are called as zona pellucida. What we are called? Zona pellucida. The zona pellucida of ovum, it is made up of lipids and glycogen. It is made up of what? The lipid and the glycogen that. This zona pellucida is covered by the another membrane that is called as corona radiata. What we call the corona radiata. See here the plasma membrane of the ovum and the zona pellucida layer between the space is there. That is called pre-vitelline space, which is the plasma membrane of the ovum and zona pellucida layer between the gap is there. That gap is called pre-vitelline space. The first membrane is called zona pellucida. The outer membrane is called as corona radiata. See here now here the one first polar body is there. What happened? This polar body, the porched out in anterior side, to form the animal pole. To form the what? Animal pole. To opposite of the animal pole, we are called as that the vegetal pole. What we are called? This is about the vegetal pole. This total information of the secondary oocyte. Now we are going to discuss about the hormonal, the control of the oogenesis. Hormonal control of the oogenesis, we will discuss that. In endocrine system, which is the gland is called the master gland, that is called as what? The pituitary gland. Actually, the pituitary gland is under the control of the hypothalamus in the brain, which is there. Hypothalamus. This hypothalamus, due to the influence of the hypothalamus to release one hormone, that is what is the GnRH, which is that hormone is the GnRH. What is the GnRH? Gonadotropic releasing hormone. Which one that? Gonadotropic releasing hormone. Due to the influence of the gonadotropic releasing hormone, it is effect on anterior part of, it is effect on the anterior part of pituitary gland or the master gland, and it is affected what? Anterior portion of the pituitary gland. When the anterior portion of the pituitary gland, there is a release of two hormone that which one that LH, another one is FSH. LH or FSH, luteinizing hormone and follicle stimulating hormones are released due to the influence of anterior part of the pituitary gland, influence of GnRH. That these LH and FSH are effect on the ovary, primary sex organ of the female reproductive system, effect on the ovaries, effect on the what? The ovaries. From the ovaries, what happened to release the two hormones, that is progesterone and estrogen. Progesterone and estrogen, which in the ovaries, progesterone and estrogen. This progesterone and estrogen, it is effect on the uterus. It is effect on the uterus. Due to the effect of the uterus, what happened? Finally, that oogenesis is going to happen. That this total about the what the oogenesis, the significance of the oogenesis, hormonal control of the oogenesis is very important. In this topic, there is a which is oogonia is a which nature, that diploid or applied, the step process oogonium, primary oocyte, the secondary oocyte, the ootids, one oogonium to release the only one ootid, three polar bodies. The ootid is a secondary oocyte. It is arrested in from metaphase 2, meiosis 2 and metaphase 2. These all are the very important for the entrance examination. Once or twice you can watch this video, then comment, then any doubt is there, keep on comment section. Thank you one and all.